The world around us, it's just filled with sound, isn't it? The rustle of leaves, maybe your favorite song, the voice of someone you care about. We hear these things, perceive them pretty much effortlessly. But if you actually stop and think about it for a second, I mean, what are sounds? They're just invisible vibrations in the air, right?
Yeah.
So how do these seemingly simple disturbances, how do they transform into the incredibly rich, complex, and, well, meaningful world of sound we actually experience inside our heads? It's really an everyday marvel, a staggering feat of biological engineering that, honestly, we often just take for granted. So today, we're diving deep into exactly that question. We're going to try and unravel some of the biological wizardry behind our hearing. And our main guide, our compass for this deep dive, is this really extraordinary tool, the Advanced Auditory Hair Cell Simulation. Now, we spent a good amount of time absorbing the source material you gave us, the All Notes 1-800-623-205, and the Auditory Hair Cell Simulation and Neural Coding document. Our mission here is pretty clear. We want to pull out the most crucial insights from these sources, the goal, to really help us all grasp how our ears convert that raw sound energy into, well, the precise electrical language our nervous system actually understands. Okay, let's try and unpack this pretty intricate process. So deep inside your skull, tucked away in the temporal bone, you've got this tiny spiral structure. Looks a bit like a snail shell, actually. That's the cochlea. It's the real control center of your inner ear. And inside this remarkable structure, you find the real unsung heroes of hearing, these incredibly specialized cells known as hair cells. Now, these aren't just passive receivers, not at all. They are biological transducers. Think of them like master alchemists, turning mechanical vibrations into the neural signals your brain can actually interpret. But the big question, the central enigma we're exploring today is how? How precisely does your body achieve this absolutely astonishing transformation? And crucially, how does the simulation help illuminate that process?
What's truly fascinating here is just the sheer elegance, but also the complexity that's embedded deep within the auditory system. It's really far from a simple mechanism, and that's precisely why this simulation, the one we're about to dig into, becomes such a powerful learning tool. It provides those crucial aha moments. It makes these abstract biological mechanisms feel tangible, observable. For anyone who's curious about the mechanics of our senses, actually seeing these intricate processes unfold dynamically in a simulated environment, it really brings home the ingenuity, the evolutionary refinement of our own biology. It's really a testament, I think, to millions of years of natural selection, designing this incredible system that lets us interact with and precisely interpret the world through sound. It's quite something.
Okay, so let's start by setting the scene. Right inside the powerhouse of our inner ear, the cochlea. We often say it's snail-shaped, and it really is. It's this beautiful, intricate spiral tucked away, protected deep within your skull. And this isn't just some empty container. It's the central arena, you could say, the main stage where the initial and arguably the most crucial processing of sound actually begins. It's a really remarkable piece of biological architecture, seems perfectly designed for its very specific, vital role. But what's actually doing the heavy lifting inside this tiny spiral?
Well, at the absolute heart of that arena, you find the hair cells. And these are just, passive detectors. They are highly specialized, incredibly delicate sensory cells. They're absolutely fundamental to our ability to hear anything at all. Their primary defining role is that of a transducer. Now, to put that maybe a bit more simply, they take mechanical energy, those tiny, tiny pressure waves that make up sound, and they convert them into electrochemical signals, and then ultimately electrical signals. It's kind of like a sophisticated microphone, taking sound waves and turning them into electrical impulses that an audio system can then process.
Okay.
But in our case, that audio system is the unbelievably complex network of our brain. These hair cells, they form the critical bridge. They transform the physical world of sound into the biochemical and electrical language of our nervous system.
Yeah.
And the precision of this conversion, the speed, the nuance, it's really quite astounding when you think about it.
So this advanced auditory hair cell simulation, it's not just a static picture then, or some dry diagram from a textbook. What's its fundamental function in showing us this complex process?
Oh, absolutely not. Not static at all. The simulation's core purpose is to offer a dynamic, living, almost interactive representation of this precise process. How those incoming sound pressure waves get meticulously transformed into what the sources really aptly call the electrical language of the nervous system. It's incredibly focused. It really zooms in with remarkable precision on the intricate, almost microscopic role that these hair cells play in that initial, absolutely vital conversion. It doesn't try to model the entire auditory pathway. That would be far too complex for any single simulation right now. Instead, it gives us an exceptionally clear, very focused, and detailed view of this single most fundamental and utterly foundational step in the entire hearing process. It's almost like putting a powerful microscope right on that very first moment where sound becomes information the brain can use.
Right. And why is simulating this specific step, this initial conversion, mechanical to electrical? Why is that so absolutely foundational to understanding hearing as a whole? Why start there? Why is it the bedrock?
Well, because, quite simply, without this initial conversion step, there is no hearing, full stop. It is the very first, utterly indispensable step in what becomes a long and complex chain of events that eventually lets our brain interpret, recognize, and respond to sound. If these mechanical vibrations aren't accurately, efficiently, and incredibly rapidly translated to electrical signals, then the brain just gets, well, nothing, no information at all.
Like silence.
Exactly. It's like trying to listen to a radio that isn't plugged in. Doesn't matter how good the radio is.
Yeah.
So understanding how these hair cells actually achieve this conversion, how they manage to differentiate and respond to vastly different frequencies, different intensities of sound, that's the absolute prerequisite. It's essential for comprehending everything else that happens further up the auditory pathway. Things like our ability to perceive pitch or understand complex speech or even figure out where a sound is coming from in space. The simulation lets us dissect that crucial, almost magical moment where raw sound energy crosses that biological threshold and becomes neural information. It really is the critical first gate.
Okay, so with that vital stage set, let's dive into the core principles, the really fascinating mechanics that the simulation brings to life so vividly. And the first one you mentioned is called tonotopic mapping. Now, for listeners who follow sensory biology, this idea, it's pretty central to how our ears work as these amazing frequency analyzers, right?
Precisely. Yeah, what this principle explains is that the cochlea isn't just some uniform structure that vibrates randomly. Not at all. Instead, it's spatially organized with incredible precision, almost like a meticulously laid out piano keyboard, as you said. It's specifically designed so that different locations along its spiral length are maximally sensitive to different frequencies or pitches. And this organization isn't just precise, it's also remarkably consistent across individuals. It forms this stable map of sound frequencies inside the ear.
Okay, so if you were to hear a really high-pitched sound, maybe like a piercing fire alarm or just a high note on a piano, where does that primarily activate inside the ear according to this map?
That specific activation would happen predominantly right at the base of the cochlea. You can think of the base as the entrance, really. The very first part of that spiral structure that incoming sound waves encounter. Structurally, this part of the cochlea is relatively narrow, and it's quite stiff compared to the other end. And it's these specific physical properties, its stiffness and its narrowness, that make it inherently vibrate most efficiently in response to high-frequency sounds.
Ah, okay.
So if you were to go into the simulation and actually input a high-frequency sound using the slider, you would clearly see the peak of the vibrations concentrated right there on the left side of the canvas. That represents the basal high-frequency region. It's almost as if the highest keys on our internal piano keyboard are positioned right there at the entrance.
And conversely, what about the really deep, resonant, low-frequency sounds? Like a rumbling bass guitar or maybe distant thunder or the lowest notes on a cello. Where do they make their main impact?
Right. So those low-frequency sounds, which have much longer wavelengths, they actually travel much further down the cochlear spiral. They go all the way to the apex. The apex is the far end of the cochlea, the very tip of that snail shell structure. And structurally, it's quite different from the base. It's significantly wider and it's much more flexible, much less stiff.
Okay.
These distinct characteristics make it perfectly suited to resonate with and respond most strongly to low-frequency sounds. So again, in the simulation, if you were to grab that frequency slider and dial it right down to a low frequency, you'd visually observe the peak of those vibrations shifting all the way over to the right side of the canvas.
Yeah.
It truly provides this dynamic spatial map of frequencies. It lets you see this abstract concept really clearly in action.
And here's where it gets really interesting, I think. The simulation isn't just telling you about tonotopic mapping. It actually makes this complex, kind of abstract idea incredibly tangible, interactive. You can literally watch this map being created as you adjust the frequency.
Precisely. That's the beauty of it. As you smoothly move that slider back and forth, the peak of the vibration dynamically travels along the displayed row of hair cells. It's almost like watching a tiny wave crest moving down this specialized biological channel with its peak landing exactly where that specific frequency is tuned to resonate most strongly.
Wow.
And if we connect this back to the bigger picture, this incredibly precise spatial organization is absolutely critical. It's fundamental for our brain to accurately distinguish between different pitches. It's the core reason we can discern, say, a flute from a violin in an orchestra, even when they play the same note loudness. Or why a busy street doesn't just sound like an undifferentiated wall of noise.
Hmm.
Your cochlea is actively physically dissecting that soundscape into its distinct frequency components, and it sends this remarkably organized, spatially coherent data stream up to your brain. This isn't just some neat organizational trick, it's the core mechanism that enables our rich perception of melody, harmony, and the overall texture of sound.
All right, so our ear has this incredibly sophisticated internal piano keyboard mapping out all the frequencies. That's amazing. But once those vibrations hit the right key on that keyboard, how does the ear actually feel those vibrations? And more importantly, turn them into something electrical, something the brain can actually understand. That brings us neatly to the second key principle, which reveals it's not just one type of hair cell doing all the work. It's a really remarkable collaboration described as the hair cell duo sensors and biological amplifiers.
That's absolutely right. Yeah, the simulation makes it very clear visually. It distinguishes between two distinct populations of these delicate hair cells. And each population plays a very different, yet equally vital and actually complementary role in the whole hearing process. Understanding this duality, this teamwork, is really key to appreciating just how sophisticated our auditory system truly is.
Okay, so first up we have the inner hair cells, the IHCs. And in the simulation, they're shown as a single, neat row of pink cells. Now, from what I understand from the sources, these are the primary sensors. They're the ones doing the direct communication with the brain. What's their specific critical job here?
The inner hair cells, yes, they are indeed the true gatekeepers of auditory information. They are essentially the only cells that transmit the actual sound information up to the brain. So when those mechanical vibrations, those microscopic sound waves we've been talking about, when they're strong enough to physically bend the tiny hair-like bundles, they're called stereocilia, on top of these IHCs, it triggers this rapid electrochemical cascade.
Okay.
This bending physically pulls open tiny ion channels that allows positively charged ions, mostly potassium and calcium, to rush into the cell. This influx depolarizes the cell, changes its electrical charge, and this depolarization, in turn, causes the cell to release chemical messengers, neurotransmitters, primarily glutamate, into the tiny gap, the synapse, between the hair cell and the nerve fiber. These neurotransmitters then bind to receptors on the dendrites of the auditory nerve fibers. And that binding is what directly sends an electrical signal, an action potential or a nerve spike, straight up the auditory nerve towards the brain. They are the direct, absolutely essential link from the mechanical world of sound vibrations to the electrical world of your central nervous system. And in terms of encoding loudness, which we'll touch on more, the sources indicate that if a sound is quiet, maybe only a few of these IHCs will fire, and only occasionally sending sparse signals.
Makes sense.
But if the sound gets loud, many IHCs are recruited, and they fire very rapidly, very frequently, signaling that increased intensity to the brain. It's a remarkably efficient and precise communication system.
Okay, so the IHCs are the direct communicators, sending that essential sound is here signal and encoding its basic intensity. But then we have the outer hair cells, the OHCs. The simulation shows these as 3 distinct rows of teal-colored cells, and these are famously labeled as biological amplifiers. Now, that's a really fascinating term. What exactly does a biological amplifier do in this context, and how on earth does it work?
Ah, yes. This is where the true microengineering genius of the cochlea really shines. This adds an absolutely astonishing layer of sensitivity and dynamic range to our hearing. The outer hair cells, unlike the IHCs, they don't directly send the primary sound signals to the brain in the same way. Instead, their critical, unique role is to physically amplify the minute mechanical vibrations before those vibrations even effectively reach the inner hair cell.
They boost the vibration itself.
Exactly.
Yeah.
They achieve this through a really unique and incredibly rapid process called electromotility. Essentially, these highly specialized cells possess a remarkable ability to rapidly and precisely change their physical length. They literally contract and elongate, like tiny pistons, in direct response to changes in their own electrical potential. This incredibly fast, active movement physically boosts the vibrations of the basilar membrane, which is the structure that both types of hair cells sit on. So imagine you're trying to hear a very, very faint sound, like maybe a whisper from across a quiet room, or the distant rustle of leaves outside on a calm day.
Yeah.
For these quiet, low-energy sounds, the OHCs actively and physically move, literally boosting those tiny mechanical vibrations within the cochlea's fluid. This active amplification makes the inner hair cells remember there are primary sensors much, much more sensitive. It effectively allows them to pick up and signal those incredibly faint sounds that would otherwise be completely imperceptible, below our threshold of hearing. Without this active amplification from the OHCs, our auditory world would be drastically different. It would largely be filled with only loud noises. All the nuances of quiet speech, the delicate notes in music, the subtle sounds of nature, they would simply vanish. This is precisely why OHC dysfunction or damage is a primary cause of many common forms of sensorineural hearing loss. It profoundly impacts our sensitivity, especially to quiet sounds.
That's a really crucial point. So just to be clear, if the OHCs weren't there, or if they were significantly damaged, say, by loud noise exposure, would we still be able to hear anything at all?
Well, yes, you would likely still be able to perceive very loud sounds. Things like a jet engine roaring overhead, a loud clap of thunder nearby, or someone shouting right at you. These intense sounds would probably still create enough raw mechanical vibration to directly stimulate the inner hair cells, even without any help from the OHC amplifiers. However, anything subtle, a normal conversation level, a soft melody, the gentle ticking of a clock, even the low-level sounds of speech would be profoundly diminished, or perhaps entirely lost. It's kind of analogous to having a sound system where the preamplifier is broken. The main power amplifier might still work, but only the very loudest inputs will actually get through and be heard.
I see.
This is exactly why OHC damage is such a significant factor in so many common types of hearing loss. It dramatically reduces sensitivity. And what's also biologically brilliant, just ingenious design, is that this amplification mechanism is actively suppressed when sounds are already loud. There's simply no biological need to amplify an already booming sound, right? And this suppression also serves a vital protective function. It helps prevent the delicate inner hair cells from being overstimulated or even damaged by excessive vibration from loud noises. It's a dynamic, self-regulating protective system.
That makes perfect sense. And it leads us really nicely into the third fundamental principle that the simulation helps us understand. This is all about how our brain actually figures out how loud something is. It's a sophisticated process described in the sources as neural coding of loudness, the brain's volume knob.
Exactly. The brain doesn't just need to know what the sound is or its pitch based on where it is on that tonotopic map. It also desperately needs to know its intensity, its volume. And this loudness information isn't encoded merely by, say, counting how many hair cells are firing, although that plays a role. It's primarily, and most dynamically, encoded by the firing rate of the auditory nerve. Think of it less like a simple light switch that's either on or off, and maybe more like Morse code. The meaning, the intensity changes dramatically based on how quickly those electrical spikes or signals are being sent. A slow, deliberate sequence of spikes means one thing, quiet, while a rapid-fire burst of spikes means something entirely different, loud.
And the simulation actually lets us see this neural output, right? It visualizes these electrical signals, which must be fantastic for getting your head around this concept.
Yes, absolutely. The neural output graph in the simulation is essentially your direct window into the language the brain is actually receiving from the ear. It meticulously visualizes these electrical signals that originate from the inner hair cells and then travel up the auditory nerve. Each individual red line you observe scrolling across that graph represents a single spike, which is technically an electrical impulse, an action potential. Now, if a sound input is quiet, you'll see a relatively low rate of these neural spikes appearing on the graph. That's because, as we discussed, only a few inner hair cells are firing, and they're firing only occasionally, sending a more relaxed signal train. But crucially, if the sound becomes loud, you'll immediately observe a dramatically high-frequency train of spikes. It looks like a rapid, dense burst of red lines flowing across the graph. This visually reflects that many IHCs are now firing, and they're firing very rapidly and intensely. The density and the speed of those spikes scrolling by are the direct visual representation of loudness being encoded.
And then, arguably, the truly brilliant part for really understanding this is the sonification feature. You mentioned that this lets you actually hear this neural activity, which just makes it so concrete, doesn't it?
Oh, it's an incredibly powerful feature for intuitive understanding. It really brings the concept to life in a way visuals alone can't quite capture. When you enable sonification, that low neural firing rate, the one indicating a quiet sound, translates into sparse, distinct clicks that you can actually hear through your speakers or headphones. It sounds almost like a very slow Geiger counter, detecting very few particles.
Oh,
But then, as you increase the loudness of the sound input in the simulation, and the neural firing rate dramatically increases on the graph, those individual clicks audibly merge together. They become a rapid, continuous crackle or static noise. You are literally hearing in real time the way your nervous system encodes volume. It transforms what could be a pretty abstract biological process into a direct, visceral auditory experience. It's quite effective.
So what does this all really mean for us, for our perception? It means the brain isn't just hearing a sound as some simple entity. It's actively, constantly deciphering this complex code of clicks and crackles, really, the frequency and intensity of electrical impulses arriving from potentially thousands of nerve fibers. It uses that code to determine precisely how loud that sound is. It's so much more intricate and dynamic than just a simple on-off switch or some static volume dial in our head. It's a highly sophisticated, real-time data stream interpretation.
Absolutely. And this elegant encoding mechanism, it naturally raises an important question. How precisely does the brain then interpret these subtle variations in firing patterns, these different rates of neural spikes arriving from different parts of the cochlea? How does it use that information to distinguish, for instance, the gentle rustle of leaves from the deafening roar of an engine? It's not simply about detecting the presence or absence of sound. It's about the brain's sophisticated ability to decode the intensity information embedded within those complex patterns of electrical impulses, integrating information across multiple auditory nerve fibers simultaneously. And that's what gives us our incredibly rich and nuanced perception of volume, from the faintest whisper to the loudest thunderclap. This precise encoding of loudness is absolutely fundamental to how we navigate and interact safely and effectively with our auditory environment every single day. It allows us to react appropriately, whether it's leaning in to hear a whisper or covering our ears from a sudden, dangerously loud noise.
Okay, so this simulation, it's clearly not just a static video or animation you passively watch. The sources emphasize it's designed more like a fully interactive lab environment. It really puts you in the driver's seat, doesn't it? It allows you to actively explore and observe these really profound principles of hearing firsthand. So tell us, how do you, as the user, actually interact directly with this advanced simulation? What can you do?
That is indeed a core strength of its design, and it's really what makes it such an effective learning tool, I think. It explicitly encourages active engagement. It really transforms you, the user, into the experimenter.
Right.
You're not just passively absorbing information, you're actively manipulating variables, maybe making predictions about what you think will happen, and then observing the real-time biological responses right there on the screen. This kind of hands-on, direct observation approach, it deepens understanding far more effectively, I believe, than merely reading about these complex processes in a book ever could. You truly get to, play around with the underlying mechanics of hearing.
Let's break down the main controls available to the user then. There's a pretty prominent play demo button right at the top. What does that do for someone maybe just getting started or perhaps wanting a quick overview?
Yeah, the play demo button is an excellent starting point, or even just a quick refresher if you've used it before. When you activate it, it initiates this automated demonstration sequence that cycles through the key features and principles we've just been discussing, highlighting them one by one. For instance, it might begin by showcasing that remarkable OHC amplification in action, specifically at quiet sound levels. Then it might seamlessly transition into demonstrating frequency sweeps across the tonotopic map, letting you observe how that peak vibration shifts smoothly across the hair cells.
Okay, sort of a guided tour.
Exactly, a guided tour. It's intelligently designed to give you quick, illustrative insights into the core functionalities without you having to manually adjust anything right away. And importantly, you can press that same button again at any point to stop the demo and immediately regain full manual control. That's incredibly useful if you want to pause and examine something specific, or when you're ready to dive deeper and start your own focused experimentation with the sliders.
Okay, then we have the two main sliders, which are arguably the real heart of the interactive experience: frequency hertz and amplitude loudness.
Yeah.
We've already touched on the frequency hertz slider quite a bit in relation to tonotopic mapping, but let's just reiterate how crucial interacting with this specific slider is for truly grasping that concept.
Absolutely. The frequency hertz slider gives you direct granular control over the pitch of the simulated sound you're feeding into the system. It allows you to sweep smoothly across a really wide range, from deep rumbling bass frequencies way up to incredibly high treble tones. And as you glide this slider back and forth, you could visually witness, in real time, the peak of the vibration dramatically shifting its position along the entire displayed row of hair cells on the main simulation canvas.
You actually see it move.
You see it move. This immediate direct visual feedback makes that previously abstract concept of tonotopic mapping incredibly concrete and intuitive. You quite literally see how different pitches precisely activate different geographical locations within the cochlea, physically solidifying that internal piano keyboard analogy we keep coming back to. It's a really powerful demonstration of the ear's fundamental frequency analysis capabilities.
And the amplitude loudness slider, this seems to be where we can truly witness those outer hair cells, those biological amplifiers in all their dynamic glory. What specific observations can a user make by playing with this control?
Yes, this slider is indeed exceptionally insightful for understanding the huge dynamic range of our hearing, and specifically the critical role of the OHCs in achieving that range. It allows you to meticulously control the intensity or the volume of the simulated sound input. Now, what's particularly compelling and really visible is that at quiet amplitude settings, when you turn the volume down low, you can directly observe the outer hair cells physically contracting and elongating. You see them moving with remarkable precision and speed. They are actively amplifying that weak mechanical signal to make it audible, strong enough for the IHCs to detect. It's like watching the silent, rapid dance of biological micromotors.
Wow.
Then, as you gradually increase the amplitude using the slider, pushing it towards loud levels, you'll observe this amplification progressively switching off. The OHCs actually suppress their active electromodal role. They become much more passive. This visually showcases their brilliant biological efficiency, as we said. There's no need to amplify an already booming sound, and also their protective function. And crucially, as you adjust this amplitude slider up and down, you'll also see the corresponding and often dramatic change in the neural firing rate displayed on the neural output graph right below. This interaction allows you to directly witness the biological volume knob in action. You see not just how the ear dynamically adjusts its own sensitivity, but also how that varying intensity information is precisely encoded in the neural signal being sent to the brain. It makes the abstract concept of dynamic range feel very real and palpable.
Okay, so beyond the hands-on controls, the simulation offers several key visual displays that act as crucial windows into this amazing process. The simulation canvas is obviously the main show where everything happens.
Exactly. The simulation canvas is the primary visual display area. It presents what the sources describe as an unrolled segment of the cochlea, almost as if you've mathematically laid it flat to get a clearer, linear view of the components. This is the central stage where all the intricate action unfolds in real time. You see the individual hair cells, both inner and outer. You observe the propagating vibrations moving along the structure. You witness the rapid ion influx, which is indicated by the cells glowing green when they activate. And you see how these events ultimately lead to the generation of those red neural spikes. It's the visual heart of the simulation, allowing you to observe the intricate, coordinated dance of all these biological components working together. Without it, you'd be missing the dynamic interaction entirely.
And then there's the neural output graph, where we talked about seeing those critical red spikes. You also mentioned that really powerful sonification feature is integrated right there with it.
That's right. The neural output graph is your direct window into what the brain is actually receiving from the inner ear, the electrical signal. It meticulously visualizes the train of electrical signals being sent up the auditory nerve pathway. Each discrete red vertical line you see scrolling across that graph marks a single spike, a single action potential fired by an auditory nerve fiber connected to an inner hair cell.
Got it.
And, yes, right next to the graph's title, there's a little speaker icon button. Clicking that allows you to enable or disable the sonification feature. And this is arguably one of the simulation's most impactful features for true comprehension, I think. It lets you actually hear the neural activity as it happens. So a low neural firing rate representing a quiet sound produces those sparse, distinct clicks. You hear kick, click, kick, gives you immediate auditory representation of how quiet sounds are sparsely encoded.
Right.
Conversely, a high neural firing rate representing a loud sound results in a continuous rapid crackle or static noise. You hear something like "kurr." This directly demonstrates the furious rapid neural firing associated with high intensity. It provides an immediate, intuitive, and deeply auditory sense of how loudness is directly translated into the pattern of neural activity over time. It's a phenomenal way to bridge that gap between abstract neuroscience concepts and something you can actually experience.
And finally, just quickly, we have the legend. Now, it might seem like just a small detail, maybe easy to overlook, but why is it so incredibly important for understanding everything we're seeing on the canvas and in the graphs?
Oh, the legend is far from a small detail. It's absolutely critical for accurately interpreting the complex visual information presented, especially on the main simulation canvas, and also for understanding the neural output graph correctly. Think of it as your essential Rosetta Stone for the specific visual language used by this simulation. It meticulously outlines the color coding system. For instance, it clearly tells you that the inner hair cells, those primary sensory cells, are consistently depicted in pink. The outer hair cells, the crucial biological amplifiers, are always represented in teal. It also explains that the ion influx, which visually signifies the moment of activation as ions flow into a cell, is shown as a temporary green glow within the cell. And of course that the neural spikes, the signals actually being sent to the brain, are always shown as those distinct red lines on the graph.
Okay, makes sense.
Without this legend, you'd be looking at a beautifully animated, but ultimately cryptic and potentially confusing display. It's absolutely essential for correctly linking the dynamic visual changes you observe on screen to the specific underlying biological components and processes they represent. It ensures you understand exactly what you're observing and why it's happening. It really demystifies the visuals and makes the whole simulation interpretable.
So we've just spent a good amount of time exploring how advanced and, frankly, remarkably insightful this auditory hair cell simulation really is. And it truly seems like a powerful tool for understanding the basics. But interestingly, the source material itself is quite open about the fact that it also has some profoundly interesting limitations. Now, these aren't necessarily shortcomings in a purely negative sense, are they? They feel more like, well, like exciting, perhaps even tantalizing avenues for future enhancements. They seem to highlight precisely where the cutting edge of auditory science and modeling might be headed next.
You've hit on a really crucial point there.
Yeah.
While the simulation is rightly described as advanced, and it does accurately model these fundamental principles of hearing with impressive detail, the sources are very clear and upfront that it still provides inevitably a simplified view of a biological system that is almost unfathomably complex in reality.
Right.
These acknowledged limitations are actually incredibly informative. They serve not only to help us appreciate the immense complexity of the auditory system beyond these core principles we've discussed, but also to clearly delineate where the frontiers of our scientific understanding and our technological modeling capabilities still lie. They effectively show us what puzzles remain to be solved and where future research and simulation efforts will undoubtedly need to focus. It's a valuable reminder of just how much more there is still to unravel about hearing.
Okay, so what's the first major limitation that's mentioned in the sources? It seems to relate specifically to the type of sound input the simulation can currently handle.
Yes, the first key limitation discussed is what the sources refer to as simplified sound input. Essentially, in its current iteration, the simulation is restricted. It can only model the cochlea's response to a single sine wave at a time. Now, for our listeners, just to clarify, a sine wave is the purest, most fundamental form of sound imaginable. It's a pure tone, like the sound you'd get from a perfectly tuned tuning fork, or maybe a very simple single electronic beep from old equipment. It's just one single unvarying frequency.
Just one note.
Exactly, just one pure note.
And why is that restriction only handling one pure tone such a significant constraint when we think about the richness and complexity of the sounds we encounter in the real world every day?
Well, because real-world sounds are almost never just single, pure sine waves. Think about it for a moment. A human voice speaking, the melody of a song, the sheer cacophony of a busy city street, or the intricate interwoven layers of sound in an orchestral piece. These are all incredibly complex auditory signals. They're composed of many, many different frequencies oscillating simultaneously, often with wildly varying amplitudes and phase relationships between them.
Right.
This fundamental limitation means the current simulation cannot simulate these more complex auditory inputs. It can't handle, for example, two simultaneous frequencies played together to form a musical chord, or the broadband energy spread across many frequencies that constitutes white noise, let alone the incredibly nuanced and rapidly changing complexity of human speech or music. A significant consequence of this limitation is that the simulation cannot demonstrate how the cochlea truly separates and analyzes complex sounds into their constituent frequency parts. This is a process sometimes called Fourier analysis, which our ears perform naturally and constantly. This is a massive limitation because our cochlea is actually incredibly adept at this frequency analysis. It essentially breaks down a complex sound, like that entire orchestra playing at once, into all its individual component frequencies along that tonotopic map. So the simulation as it stands can't show you how your ear manages to disentangle the sound of the individual instruments in that orchestra, or perhaps more relevantly, how you can usually pick out a single familiar voice speaking amidst the chatter and clatter of a crowded, noisy room. And this limitation, it directly raises a really important question, doesn't it? If this simulation, which accurately models the core mechanics we've discussed, can only process one pure tone at a time, then how on earth does our incredibly sophisticated biological ear cope with the absolute cacophony of everyday life? It really highlights that beyond this foundational single-frequency processing, there's just immense and still not fully understood complexity in how our auditory system performs highly sophisticated signal processing to decompose, filter, and ultimately reconstruct our rich, coherent soundscape from the messy input it receives.
Okay, that makes sense. Another area where the simulation, according to the sources, admits a fascinating limitation is the lack of hearing loss modeling. So it currently can't simulate how different types of common hearing damage might actually affect the process.
That's absolutely correct, and it's a very significant point, especially if you think about the potential educational or even clinical applications. The current versions does not allow for the simulation of common forms of hearing damage or auditory pathology. And this significantly restricts its utility in a really crucial area for both research and clinical understanding.
How so? What could you do if it did model damage?
Well, for instance, remember how we talked about the outer hair cells acting as those vital biological amplifiers? Absolutely essential for hearing quiet sounds.
Right, the amplifiers.
So imagine if you could go into the simulation and selectively damage or remove a specific patch of those OHCs in a certain frequency region. If you could do that, you would then be able to directly observe the immediate, localized impact on the system's ability to perceive faint noises in that frequency range. You could visually see the reduction in their active amplifying movement, and you could see the corresponding decrease in the neural output for quiet sounds, specifically in that region.
I see.
Similarly, the current simulation cannot simulate, say, removing entire sections of inner hair cells, perhaps mimicking damage from extreme noise exposure or certain medications. Doing so could create dead spots or regions where certain frequencies simply cannot be detected at all, which would visually simulate the kind of localized, frequency-specific hearing loss that many people experience.
So without this capability to model damage, it's not really usable as, say, a diagnostic tool, or even as an educational tool for truly understanding the progression of hearing loss, or
Right.
maybe visualizing the potential effects of treatments like hearing aids.
Exactly. Its current inability to model various forms of damage significantly restricts its utility as a tool for understanding exactly how different types of damage affect hearing sensitivity across frequencies. It can't really show how specific pathologies like presbycusis, which is common age-related hearing loss, or noise-induced hearing loss, might progress over time. And importantly, it limits its use for exploring or visualizing the potential impact of various interventions. Think about hearing aids, which selectively amplify certain frequencies, or cochlear implants, which bypass the hair cells altogether, or even potential future treatments like novel gene therapies aimed at regenerating hair cells. You can't really model their effects at this fundamental level within the current simulation. While it's absolutely fantastic for illustrating the fundamental principles in a healthy, idealized ear, it stops short of being able to apply those principles directly to the complexities of real-world medical scenarios and the significant challenges posed by impaired hearing. Just imagine the transformative potential for such a feature in future versions, though. Not just for educating future audiologists, doctors, or hearing researchers, but potentially even for diagnostic visualization, or perhaps helping patients and clinicians truly visualize the specific functional effects of their particular type or pattern of hearing loss. It really opens up an entirely new dimension of potential practical application.
Okay, and finally, there's one more limitation mentioned, which sounds quite technical. The absence of efferent pathway simulation. What exactly does that term mean, and why is its absence considered a limitation for understanding hearing?
Right, this refers to a less commonly discussed, but actually incredibly important aspect of the overall auditory system, the efferent pathway. We typically tend to think of hearing as a purely one-way street, right?
Oh.
Sound comes into the ear, it gets processed, travels up the auditory nerve to the brain, and then we perceive it. End of story.
Yeah, that's how I usually think of it.
But the efferent pathway represents the direct anatomical and functional mechanism by which the brain actually sends signals back down, descending from higher auditory centers to the cochlea itself. It actively influences how the cochlea processes incoming sound even before it gets sent up. It's a sophisticated top-down control system.
Wait, the brain talks back to the ear. Why? What's the purpose of that kind of feedback loop?
Well, primarily the brain sends these efferent signals down to actively modulate the cochlea's sensitivity and help filter the incoming auditory information. And interestingly, it does this largely by controlling those very same Outer hair cell amplifiers we spent so much time discussing earlier.
Ah, the OHCs again.
Yes. The brain can essentially tell the OHCs in specific frequency regions to either increase or decrease their level of amplification. It might do this based on what sound it expects to hear next, or perhaps more commonly, based on what sound it wants to focus on amidst a complex and noisy background environment. What's truly fascinating here, and often underappreciated, is that hearing isn't just this passive bottom-up one-way street from the ear to the brain. The brain is actively, intelligently fine-tuning what we hear at the earliest possible stage, right inside the cochlea. And this missing piece in the simulation, this efferent control, is absolutely crucial for understanding how we manage to do things like filter out distractions and selectively focus on specific sounds in our environment.
So in real life, this efferent pathway, this brain-to-ear communication, this is the very mechanism that allows us to handle something like the classic cocktail party effect. You know, managing somehow to follow your friend's conversation in a really loud, busy coffee shop, despite all the background clatter, the music, other people talking all around you.
Precisely. That's the quintessential example, the perfect illustration of the efferent system in action. Your brain, using this efferent pathway, can actively dampen the amplification provided by the OHCs for certain background noises, perhaps in frequency ranges it deems irrelevant, while simultaneously enhancing the sensitivity, boosting the OHC amplification in the specific frequency range occupied by your friend's voice.
That's incredible.
It is. It's an incredible feat of neural control, enabling selective auditory attention, and it's fundamentally driven by this top-down feedback loop. So, since the current simulation does not incorporate this crucial top-down control from the brain back to the ear, it simply cannot simulate these essential, everyday aspects of auditory processing. Things like selective attention in noise, active noise reduction strategies, or even how our internal expectations or focus can subtly shape our perception of the sounds around us. It's a major part of how we successfully make sense of our incredibly noisy and complex auditory world. And its absence in the simulation really highlights just how much more there is to model if we want to truly capture the full complexity and adaptability of human hearing.
Okay, so the overall takeaway here regarding these limitations seems to be that while this advanced auditory hair cell simulation is clearly brilliant, maybe even revolutionary, for demonstrating the core mechanics of peripheral auditory processing, what happens right there in the cochlea,
Yes.
it ultimately provides a simplified, albeit incredibly valuable and foundational view of the whole hearing process.
Exactly. That sums it up perfectly. It effectively illustrates the fundamental physics and the intricate biology of sound transduction right at the level of the cochlea with remarkable clarity. However, it still largely omits, by necessity perhaps at this stage, the incredibly intricate processes involved in complex sound decomposition, how we really discern all the individual elements within a rich soundscape. It omits the direct, observable impact of various types of hearing damage on auditory function. And crucially, it omits the brain's active, dynamic, and essential top-down control of what we ultimately perceive. It's an indispensable foundational stepping stone, no doubt about it. But looking at its limitations also powerfully shows us just how much more there is to unravel, understand, and eventually model about this truly incredible, adaptive, and constantly working sensory system. #outro.
And there you have it. That was a truly deep dive into some of the hidden secrets of sound, all viewed through the microscopic lens of the advanced auditory hair cell simulation. We've peeled back some layers today, hopefully, to better understand the cochlea's ingenious internal piano keyboard, that tonotopic mapping where specific frequencies activate precise locations along its length. We've uncovered the critical dual roles of the inner hair cells, our primary sensors sending the signals, and the outer hair cells, those remarkable biological amplifiers, whose incredibly precise movements literally allow us to hear a whisper. And we've peered into how the brain decodes loudness, not with a simple switch, but through the complex firing rate of the auditory nerve, translating volume into a rapid-fire code of electrical spikes. Now, for you, our curious listener, we genuinely hope this deep dive has offered maybe a bit of a shortcut, a clearer path to understanding some truly complex biological engineering. It's really all about those aha moments, isn't it? Seeing how those tiny, silent, absolutely ceaseless processes happening deep within your ear translate directly into the vibrant, meaningful, and incredibly dynamic world of sound that you experience every single second of every single day. So what does this all mean for you? Well, I think it means the world of sound all around you isn't just something you passively receive. It's something being actively, meticulously, and dynamically translated by an unbelievably sophisticated, tiny biological marvel. It's constantly at work making profound sense of your entire auditory environment. It's truly quite astounding when you really start to think about it, isn't it?
It really is. And, while we've peeled back some genuinely fascinating layers of this incredible simulation today, revealing the sheer genius of our peripheral auditory system, perhaps it leaves us with an even more profound realization that our ears and our hearing system as a whole are not just passive receivers at all. They are active, highly intelligent systems, constantly adapting, interpreting, maybe even anticipating the world around us based on context and experience. This deep dive, especially in the simulation's current limits, really invites us to consider the next frontier, doesn't it? Just imagine the possibilities when we can finally fully unravel how the brain truly controls what we hear, how it actively shapes our very perception of sound through those top-down different pathways we discussed. The next generation of these simulations, ones that can model complex, multi-layered, realistic soundscapes, ones that can incorporate diverse forms of hearing loss with nuanced accuracy, and ones that crucially include that intricate brain-to-ear feedback loop. They will allow us to explore the true depths of auditory processing in ways we can only currently glimpse. It might even unlock entirely new avenues for restoring, or perhaps even enhancing, hearing in ways we can only just begin to properly imagine today. The journey into understanding the full symphony of sound and how we perceive it, it feels like it's really far from over. There's so much more to discover.
Okay, so that was, we have to acknowledge a few robots. Well, we have to acknowledge all the research done for the robots to actually able to make these simulations. But yeah, it's mainly Gemini 2.5 Pro and the Claude Sonnet 3.7 and 4 that made the simulation. We obviously have the voices. If you're not, if you haven't heard the voices before, this is the Notebook LM by Google. Go check it out. And yeah, but forgot to mention, go check out the website. Everything we do eventually appears on BioniChaos.com. And let us know what you think. Also like, subscribe, unsubscribe, you know what to do. But mainly go check out the simulations.